വെൽക്കം ടു വെറ്ററിനറി. സോ നമ്മൾ രണ്ടാമത്തെ ചാപ്റ്ററാണ് ഇപ്പോൾ ബയോകെമിസ്ട്രി ഡിസ്കസ് ചെയ്യാൻ പോകുന്നത്, ദ അമിനോ ആസിഡ് ആൻഡ് പ്രോട്ടീൻസ്. സോ ഫസ്റ്റ്, വാട്ട് ഈസ് അമിനോ ആസിഡ്? We know that this is the basic structure of an amino acid. Amino acid means this compound contain both amino group and acid group, that is carboxylic group. And the amino acids are also called as, normally in our body the amino acids are in alpha amino acids because the amino group is attached to the same carbon atom to which carboxylic atom is attached. This is carboxylic group, and so this is alpha carbon. So amino group is attached to the alpha carbon, so this is alpha amino acid. Next, classification of amino acids. Amino acids are mainly classified based on various under various headings. That is, first one, based on chemical structure. And this include that is aliphatic, when the amino acid in a linear chain, and in a chain with or without branches. Then aromatic amino acid containing aromatic ring like tryptophan, tyrosine. And next, sulfur containing amino acid like cysteine, methionine. Next, hydroxyl amino acid, that is OH group amino acid with OH group, that is threonine, serine. Next, amino acids with the amino acid with the amide group. Example are asparagine and glutamine. Then next, charged amino acids. And next, amino acid, which is proline. This is based on chemical structure. Next one is based on charged. Based on charge, that is whether it contains COO minus or NH4 plus. These are charge bearing molecules or components of an amino acid. So based on charge or based on charge producing properties, amino acids are classified into a acidic, B basic, and C neutral. And next classification is based on polarity, that is polar amino acid, which is hydrophilic. And next one is non-polar amino acid, which is hydrophobic. And next classification is based on the metabolic status. That is whether it is glucogenic, that is whether these amino acids give a glucogenic product or ketogenic or both glucogenic and ketogenic. Another classification of amino acid is based on nutritional requirement. Based on whether we have to supply these kinds of amino acids to our diet or body has whether body has the ability to produce it. It is classified into essential or indispensable and non-essential. So what is essential? Amino acid which are not synthesized in the body and therefore have to be supplied through diet are called essential amino acids. That is, these kinds of amino acids are not synthesized in the body and have to be supplied through diet. These are essential amino acids, and there is 10 essential amino acids. Examples are methionine, 2, arginine, 3, threonine, 4, tryptophan, 5, valine, 6, isoleucine, 7, leucine, 8, phenylalanine, 9, histidine, and 10, lysine. So these are the essential amino acids. There is acronym called as MAT. Then like this is M for methionine, A for arginine, T for threonine, then T for tryptophan, V for valine, I for isoleucine, L for leucine, H phenylalanine, P for phenylalanine, H for histidine, and L for lysine. MAT will fly. You can use this acronym for remember these essential amino acids, and remember that the definition for the essential amino acid carry 1 mark and example carry 2 marks. So try to remember essential amino acids. Then about the importance, then importance of essential amino acid are, we know that there are in total 20 total there are 20 amino acids present in our body, and out of this 10 are essential. So these 20 amino acids are needed, whole these 20 amino acids are needed for protein synthesis. So out of which 10 are essential, that is to be supplied through the diet. So if any of these essential amino acids get absence in the diet, or absence of any one or more essential amino acids in the diet impair protein synthesis, which leads to negative nitrogen balance. So absence of any one or more essential amino acid which result in what? Impaired protein synthesis. Impaired protein synthesis, which result in negative nitrogen balance. Negative nitrogen balance. So these essential amino acids should be provided through the diet. If our diet do not contain all of these essential amino acids, or absence of even any one essential amino acid, can impair protein synthesis and can cause a negative nitrogen balance. Then also when the proteins lack one or more essential amino acid, they do not promote proper growth and maintenance of body tissue. So this lack of any one or more essential amino acids do not promote proper growth and maintenance of body tissue. Next, non-essential amino acid. Non-essential amino acids are the amino acid that can be synthesized in the body and are not required and are not required in the diet. So non-essential എന്ന് പറഞ്ഞാൽ നമുക്ക് ബോഡി തന്നെ സ്വയം പ്രൊഡ്യൂസ് ചെയ്യാൻ പറ്റും amino acids, so we don't have to provide through diet. And examples are glycine, alanine, serine, cysteine, tyrosine, aspartic acid, glutamic acid, asparagine, glutamine, proline. And there is one more class, that is semi-essential amino acids. What is semi-essential amino acids? Among the essential amino acid, the arginine and histidine are called semi-essential amino acids. So examples are arginine and histidine. And why? As they are essential in the diet of children, pregnant and lactating women, but are not essential in the diet of normal adult, because these have to be provided through diet. For whom? For children, pregnant and lactating women. Why? എന്തുകൊണ്ട് ഇവർക്ക് മാത്രം ഫുഡ് ഡയറ്റിലൂടെ പ്രൊവൈഡ് ചെയ്യേണ്ടി വരുന്നത്? ബിക്കോസ് these semi-essential amino acids are not synthesized in sufficient quantities in these physiological groups. That's why we have to provide these amino acids, that is arginine and histidine, through diet for children, pregnant and lactating women. But these are not essential for not essential in the diet of normal adult. So now let's discuss about some of the properties of amino acid. First, the charged properties of amino acids. Amino acids are charged particles. Why? Because amino acids contains acidic COH and basic NH2 group. So based on the pH of its surrounding medium, it can act as cations and anions. So they are charged particles. And about the amphoteric nature, next is the amphoteric nature of amino acid. That is, amino acids are amphoteric because they behave as both acid and base. The parameter why? Amphoteric means amino acid behave as both acid and base. In which all condition it acts as acid, behavior as an acid. In the presence of base, amino acid can donate a proton, thus act as acid. That is, normally we know structure of amino acid is COOH, C and H2, H. So in the presence of base in the presence, this COOH group will donate an H plus. This H plus is donated by the COOH group. So it has act as an acid in basic medium or in the presence of base. And in the situation where it behaves as a base, in the presence of an acid, that is in an acidic medium, amino acid can accept a proton and thus act as a base, that is COOH, CH, NH2. If there is any acid is present, this acid will donate H plus and this amino acid will accept, or the NH2 group will accept this H plus to form this act as a base. Next property we are going to discuss is the isoelectric pH of an amino acid. So, in simple words, isoelectric pH is the pH at which amino acid exists as neutral Zwitter ions. That is pH to which amino acid exists as neutral Zwitter ions. That is, neutral Zwitter ion is dipolar ion. That is, at isoelectric pH, amino acids possess equal number of positive and negative charges, hence bear no net charge. That is, at pi, net charge on amino acid equal to 0. So, since there is no net charge, these amino acids do not move in an external electric field at its isoelectric point, isoelectric pH. Do not move in an electric field. These are few points that we have to remember about isoelectric pH. And isoelectric pH is varied for various amino acids. So, at isoelectric pH, it exists as Zwitter ionic form, that is COO minus and NH2 plus. Since it contains both positive and negative charge, but both in equal amount, so net charge to 0, but in Zwitter ionic form. Okay. So, the next topic is the peptide bond. What is peptide bond? Peptide bonds are anhydride covalent bond formed between a carboxylic group of an amino acid and an amino group of succeeding amino acid. That is, first the amino acid carboxylic group, second amino acid amino group കൂടെ കമ്പൈൻ ചെയ്യുമ്പോൾ ഉണ്ടാവുന്ന bond between two amino acids is the peptide bond, and this is an anhydrate bond because a molecule of water is released during the formation of this bond, and this is a strong covalent bond. Strong covalent bond. So what is the importance of this peptide bond? Peptide bond helps in the polymerization of amino acid to form peptides, which ranges from oligopeptides to polypeptides and proteins. So what is this? These are responsible for polymerization of amino acid to form peptides and proteins. So, what are the properties or features of peptide bond? Peptide bond are strong covalent bond. So the features of peptide bonds are, these are strong covalent bond. Strong covalent bond. Next is that it is an anhydrate bond. Anhydrate bond. Next, they are amide. There are amide linkages. Then they exist in trans configuration. Generally exist in trans configuration, but exception is the in proline, the peptide bond, it has a cis configuration. Cis form of peptide bond present in proline. Next, it has partial double bond in nature. Partial double bond in nature. That is, it exists. Normally we know that this is the structure. No? It also exists as this bond, partial double bond, by transferring this bond to here. Okay. Then next is all the atoms that are carbon, nitrogen, oxygen and hydrogen around this peptide bond are coplanar. Then next, during denaturation, peptide bonds are not affected. Because what may be the reason? Because peptide bonds are strong covalent bonds. Covalent bond. So these are the properties of peptide bond. That is, it is a strong covalent bond. Because of this strong nature, it is not affected during denaturation. Then it is an anhydride bond. It is an amide linkage into a generally existing trans configuration except in proline, where it appears in cis configuration. Then it has partial double bond in nature. Then all atoms, that is carbon, nitrogen, oxygen and hydrogen, are coplanar. Okay, this are what is the importance of peptide bond? Peptide bond are responsible for the polymerization of amino acid to form peptides and proteins. These are all about the peptide bond. So now we discuss about the peptide bond. Then now we are going to discuss about some physiologically or biologically important peptides. That is, physiologically or biologically important peptides. First one is the carnosine and serine. These are dipeptides. Carnosine, it is made up of beta alanine and histidine. And anserine is a derivative of carnosine. And these two are present in muscles. These are muscle peptides, carnosine and anserine. Then next important peptide is glutathione. Hope you all hear about this glutathione. It is also called as gamma glutamyl cystinyl glycine and abbreviated as GSH. It is a tripeptide. It is made up of what? It is in the name itself. Gamma glutamic acid, then cysteine, next glycine. So glutathione is a tripeptide made up of gamma glutamic acid, cysteine and glycine. It is present in erythrocyte in large amount, and it is a powerful reducing agent and is involved in various reduction reactions in body. That is powerful reducing agent. Next, reduced form of glutathione is biologically active and is a powerful reducing agent requiring for many reduction reactions. GSH, that is glutathione, is involved in antioxidation of toxic oxidants like hydrogen peroxide and superoxides by its peroxidase activity. It contains peroxidase activity. So it acts as involved in antioxidation of what? Of oxidants like hydrogen peroxide and superoxides. And next, glutathione is required for intestinal absorption of iron by converting Fe3 plus to Fe2 plus. Convert Fe3 plus to Fe2 plus and facilitate iron absorption. Next, glutathione is required for the reconversion of methemoglobin to hemoglobin. Helps in reconversion of methemoglobin containing Fe3 plus to hemoglobin containing Fe2 plus. So first peptide is carnosine and anserin. Second one is glutathione. Next biologically important peptide is thyrotropin releasing hormone, that is TRH. We know that it is a tripeptide. It is a tripeptide. Then it is secreted by hypothalamus. And stimulate the pituitary gland to release thyrotropin stimulating hormone. Stimulate pituitary to release what? Thyroid stimulating hormone. Next is the fourth important peptides are enkephalin. It is pentapeptide. It is a neurotransmitter. It is pentapeptide. It is neurotransmitter. It inhibits pain sensation. Enkephalin inhibits pain sensation. It is a protein that is tripeptide neurotransmitter. Next important one is oxytocin. It is a nine peptide linkages. a hormone released by posterior pituitary, and it is required for smooth muscle contraction, and water reabsorption. These are the functions of oxytocin. Next, sixth one, it is bradykinin. It also is a nanopeptide. It is a vasodilator. Next, seventh one, it is a calnidin. It is a decapeptide, 10 peptide bonds. Peptide, it's also a vasodilator. Next, eighth one, it is an angiotensins. It contains angiotensin 2, which is an octapeptide. It is derived from angiotensin 1. It is a decapeptide. Angiotensin 2 is a hypertensive peptide, stimulate the release of aldosterone from adrenal glands. And next one is glucagon. It is an hormone produced by pancreas. Next is the gramicidin. Actinomycin, which is antibiotics. Then gastrin, secreting like local hormones or GI hormones. Then insulin. So these are biologically important peptides. First one is carnosine and anthocyanin. Second one glutathione. Third one thyrotropin releasing hormone, enkephalin, oxytocin, bradykinin, gliadin, angiotensins, glucagon, gramicidin, actinomycin, that is antibiotics, then gastrin secreting, then insulin. So next we are going to discuss about the proteins. So there are many number of proteins. So we classify these proteins mainly based on three classifications. That is, one, based on chemical composition. That is, under this is simple proteins which contain only one type of amino acid. Only one type of amino acid. That include serum, albumin, then globulin, keratin. And in globulins, immunoglobulins are an exception. It is not simple proteins. Then next is conjugated protein. That is composed of more than one type of amino acids. Examples: egg, albumin, hemoglobin, casein, etc. Next is derived proteins, including a protein which derived from another protein, that is gelatin, derived from collagen, derived from collagen, proteases and peptides. And next type of classification is based on shape. Based on shape, it includes one globular, and under these examples are hemoglobin, albumin, and various enzymes. And second shape അല്ലെങ്കിൽ form of proteins are fibrous form, which include keratin, collagen, elastin. Next, third classification is based on biological function. Biological function, which include one catalytic proteins. They are enzymes. All enzymes are proteins in nature. All enzymes are proteins except ribosomes. They are RNA. Ribosomes are RNA in nature. Next are defense proteins, immunoglobulins. Next are structural proteins. Structural proteins. Example: keratin. Keratin present in hair and nail. Then collagen present in muscles. Next one, 4 hormonal proteins. They include growth hormone, insulin, etc. Next, fifth type or fifth one is contractile proteins. They are actin, myosin, tropomyosin, etc. Next, transport proteins. Serum albumin, it is a transport protein which carries what? Bilirubin. Serum albumin carries bilirubin. Fatty acids, bilirubin, fatty acids, etc. Like that, transferrin protein. Transferrin is a protein which carry iron. So next, seventh type is storage proteins. They are ferritin which store iron, and next type is visual proteins. Example are rhodopsin and iodopsin, present in retina of eye. Then next type is membrane proteins. Membrane proteins. Examples: sodium potassium pump or sodium potassium ATPase. Next type is hemostatic proteins, which include fibrinogen, prothrombin, etc. Next type are buffer proteins. Buffer proteins that include plasma proteins, hemoglobin. Next, respiratory proteins, which include hemoglobin and myoglobin. Then there are certain receptor proteins like insulin receptor, glucagon receptor, steroid hormone receptor. Then certain genetic proteins like histones, various transcription and translation factors, etc. So based on the biological functions, proteins are catalytic protein, defense protein, structural protein, hormonal proteins, contractile proteins, transport proteins, storage protein, visual proteins, membrane proteins, hemostatic proteins, buffer proteins, respiratory proteins, receptor proteins, and genetic proteins. So now we are going to discuss about the protein structures. So the structural organization of the proteins. The protein exhibit 4 different levels of structural organization, that is primary organization, primary structure. tertiary structure, quaternary structure. So first moving to primary structure. Primary structure, in simple words, the primary structure indicates the number and sequence of amino acid residues from N-terminal to C-terminal. That is, it is the sequential arrangement of amino acids from N-terminal to C-terminal, and it is in a linear chain arrangement. Amino acid are arranged in a linear chain, and in this only peptide bonds are present, and this maintains primary structure. So primary structure is not affected by denaturation, since they are made up of strong covalent peptide bond. And what is the importance of this primary structure? We know that primary structure of protein, that is specific sequence and number of amino acids, determine the higher level of the protein structure. That is importance. Primary structure determines the higher levels of protein structure. And a slight change in primary structure of proteins may result in loss of biological activity of protein, like modification of a single amino acid residue as a mutation. That is, if any mutation changes the primary structure, it changes the normal higher level of protein structure, that is tertiary or quaternary level. So disruptions or abnormality in this tertiary or quaternary structure of protein will result in loss of biological activity. So a slight change in primary structure may result in loss of biological activity. this is all about the primary structure. So next we are going to discuss about the secondary structure of proteins. So what is the secondary structure? Secondary structure is the twisting and folding of the polypeptide chain results in secondary structure. So the linear chain in the primary structure is twisted and folded to form the secondary structures. And the different secondary structures include alpha helix, beta sheets, bends, loops, and disordered regions. Normally, alpha helix and beta sheets are found in same proteins. And examples are in hemoglobin. Majority of ordered structure is alpha helix and rest is beta helix. Both are present in HB. But there is an exception, that is in keratin. Keratin have exclusively secondary structure alpha helix. So keratin is also called alpha keratin. So exclusively keratin is the alpha, which have only alpha helix structure. Then about alpha helix. Alpha helix is the most common secondary structure. It is found in proteins seen in overall hemoglobin, myoglobin, alpha keratin, then collagen, etc. You can see alpha helix in hemoglobin, myoglobin, alpha keratin, collagen, etc. And alpha helix is right-handed coiled structure. It is right-handed coiled structure. And it is stabilized extensively by hydrogen bonding. Stabilized extensively by hydrogen bonding. Then these hydrogen bonds are parallel to alpha helix, and the hydrogen bonds are individually weak but collectively large number of hydrogen bonds maintain the helical structure in the stable form. That is, alpha helix has large number of hydrogen bonds. Individually, when we look, hydrogen bond are not as much as strong as the peptide bond, but due to large number of these hydrogen bonds, these hydrogen bonds give the maximum stability for this alpha helix structure. And protein hydroxyproline, glycine are considered as helix destabilizing amino acids. Why? Because it breaks helix as they disrupt the formation of alpha helix, producing a bend. So what helix destabilizing amino acids are the which are the proline, hydroxyproline, glycine. It is also called helix breakers. helix breakers. And certain amino acids like alanine and cysteine promote helix formation. Okay. Then next is beta sheet. So next is beta sheet. Beta sheets are extended structure where bonding polypeptide chains are arranged in the form of sheet, in which bonding polypeptide chains are arranged in sheets or in a pleated or zigzag pattern. Next, these beta sheets as stabilized by hydrogen bonds like alpha helix, and it is formed between H bond as formed between CO and NH of the peptide bonds of adjacent segment of same polypeptide adjacent segments CO and NH. In between these CO and NH of adjacent segments of same polypeptide, hydrogen bond is formed, which helps in holding the chain side by side. And the hydrogen bonds are the hydrogen bonds are perpendicular to the polypeptide chains. Hydrogen bonds are perpendicular to polypeptide chain. Next, parallel or anti-parallel beta sheets are there. That is, beta sheet structure can be parallel or anti-parallel depending on the direction of bonding chains. So, parallel beta sheet, that is, bonding chains run in same direction. Bonding chains run in same direction. Example is flavonoxide. Next is anti-parallel beta sheet. They are bonding chains in opposite direction. of X. Examples are fibroid of silk. Fibroid of silk is the example of antiparallel beta chain. So next we discuss about the tertiary structure. And tertiary structure of protein refers to the further folding of secondary structure of polypeptide chain giving the compound 3D conformation. That is, tertiary structure gives compound 3D conformation for a protein. Then tertiary structure explains tertiary structure explains the spatial relationship of amino acids which are far apart from each other in primary structure but brought closer as a result of folding. So first of all, the primary structure twisted and folded to become secondary structure. These are again folded to give a 3D configuration. So in the amino acids which were far apart in primary structure now are come closer together by folding. So these amino acids has a different spatial arrangements than in primary structures. So in tertiary structure the tertiary structure result in the formation of domains. Result in the formation of domains. Domains. What is domains? Domains are structurally connected but functionally independent units of protein that perform a particular function. Next, bonds present in tertiary structures are hydrogen bond when there was force. Ionic bonds, hydrophobic bonds, This is about the tertiary structure. Next, about the quaternary structure. The quaternary structures are present in proteins having more than one polypeptide chain, that is oligomeric proteins. So such oligomeric proteins show one more level of higher structure, that is the quaternary structure. So present in oligomeric proteins, that is proteins having more than one polypeptide chain. Next, the quaternary structure refers to spatial arrangement of the subunit of an oligomeric protein. So, quaternary structure refers to spatial arrangement of the subunits of the oligomeric protein, and bonds present are weak covalent bonds, which includes hydrogen bonds, van der Waals force, ionic bonds, hydrophobic bonds, weak covalent disulfide bonds. Okay, this is about the quaternary structure. So we finish about the structure of the protein. Now about the denaturation of protein. What is denaturation of protein? It is a process of disorganization of native protein structure. That is, it is process of disorganization of native protein structure, and it involves what all structures undergo denaturation. Involves loss of secondary, tertiary and quaternary structure without breaking the primary structure. Okay. Then this denaturation goes denaturation cause the unfolding of native 3D form. Proteins. Then why this denaturation causes loss of secondary, tertiary, quaternary, and why not in primary? Because we know in higher structures like higher structures, that is secondary, tertiary, and quaternary, contains are maintained by weak non-covalent bonds. Thus, this can be easily disrupted by a variety of physical and chemical agents. That's why secondary, tertiary, and quaternary structures undergo denaturation, whereas primary structure of proteins not easily broken by because they are maintained by strong covalent bond. And biological relevance of this denaturation is that proteins are active only in their 3D form. Since 3D form is lost during denaturation, proteins lose their biological activity. That is, proteins have biological activity in their 3D form. So, denaturation causes loss of 3D form, thereby loss of biological activity. So this is about the denaturation of proteins. So next topic is the isoelectric pH or pI of proteins. We already discussed about the isoelectric point of an amino acids. This is equally applicable to proteins, that is isoelectric pH, that is pI of a protein is the pH at which proteins exist as sweeter ions carrying equal number of positive and negative charges and properties. Present at isolated pH, net charge is 0 because it contains equal amount of positive and negative charges. Net charge will be 0. So at pI, protein does not move in electric field. Then at pH less than pI, proteins exist as cation form. At pH less than isoelectric pH means the surrounding medium is acidic or there is presence of an acid. So this protein accept protein from the acidic medium, thus it becomes cationic, thus it acts as cation. And at pH greater than pI, proteins exist as amino form. That is, when pH is greater, it means surrounding medium is basic. So bases takes up proton from this amino acids. So C carboxyl group loses an H plus forming COO minus. That thus protein exists in negative or anionic form. Next property is that proteins have least buffering capacity and viscosity at isoelectric point. Proteins have least buffering capacity and viscosity. So proteins have least solubility and maximum precipitability at isoelectric pH. So proteins have least buffering capacity and viscosity at isoelectric pH, and proteins have least solubility and maximum precipitability at isoelectric pH. So these are the features or properties of proteins at its isoelectric point or isoelectric pH. So these are all about the chapter. ഇവിടെ ഏറ്റവും ഇമ്പോർട്ടൻ്റ് ആയിട്ട് അല്ലെങ്കിൽ റിപ്പീറ്റഡ് ക്വസ്റ്റ്യൻസ് മാത്രമേ ഇപ്പോൾ ഞാൻ ഡിസ്കസ് ചെയ്തിട്ടുള്ളൂ. സോ ക്ലാസിഫിക്കേഷൻ ഓഫ് പ്രോട്ടീൻസ് ഇമ്പോർട്ടൻ്റ് ആണ്. അതേപോലെ സ്ട്രക്ചർ ഓഫ് ക്ലാസിഫിക്കേഷൻ ഓഫ് അമിനോ ആസിഡ് and examples, definition of essential amino acid, structure of proteins, denaturation, then this isoelectric pH. These all are repeated questions, so please have please read from these topics and study main important points from these topics. So I hope you all understand this second chapter, that is the amino acids and proteins. So thank you.